Hello, everyone. Good morning, good evening, and good afternoon to all of you who have joined us today from all across the world. We are elated to have you all. It gives us immense pleasure to invite all of you to this webinar, which is a special one because we are going to focus on how to ace a poster presentation. Poster presentation being one of the key components of a student's life, a researcher's life. It's always there in the journey, right? We are also very excited because we are almost towards the start of the Omics Research Symposium, which is going to happen on March 31st and April 1st. This two-day symposium is going to be a mega event where students, faculties, industrial experts, and scientists from different backgrounds, from different organizations will assemble and come together, discuss ideas, strategies, and results of all their hard works throughout the past year, and also brainstorm about other opportunities and exciting events coming up in the future. So this is something which you don't want to miss at all, for you will be able to gain so much information, experience, and knowledge from this particular event. The two-day event has been divided, or you can say categorized, into different components, namely bioinformatics education and training, which will be highlighted and covered on the first day, research, cutting edge science, and industrial applications of bioinformatics being the other two categories in which we have divided the symposium, which will be better highlighted on the second day, that is on April the 1st. During this symposium, there will also be a poster presentation competition where students and researchers from different backgrounds, from different universities, different institutions, and different parts of the world will showcase their research, their projects, their hard work in the form of a poster, which will be evaluated by our coveted panel of judges who are from different academic institutions, different industries, and different research organizations with loads of experience in both doing cutting edge science as well as training students. So this is one of the greatest opportunity that you will ever come across to get feedback, to learn what you can do from here, what steps would take you to the pinnacle of success which you dream about, and ultimately realize your dream. So if you haven't already, I would recommend you to register for the symposium. It's completely free to be a listener, to listen to the talks. There are other opportunities as well, which I will highlight later. And you will also get to know about in today's webinar. So before we begin with today's session, I would request all of you to kindly mute yourself. Please maintain the proper Zoom decorums. Hosts, if you can please mute yourself so that we have peaceful conversation. Right, thank you. Meanwhile, while we are waiting for the start of today's session, we are waiting for a few more minutes so that everybody can join in. We would like to know about your background, what you are currently studying, where are you from, what are your interests, and why did you choose bioinformatics, or why does bioinformatics seem interesting to you? What does it appeal to you, and where do you see yourself with bioinformatics in the near future? So please use the chat section to write about it because it would help us connect with you better. Meanwhile, I will keep briefing you about the symposium so that you can know about it better. And then we will begin with today's session. And I am waiting for all of you in the chat section to know about you all. And finally, I would like to remind you that the registrations for the symposium are still open. So please make use of this time to get yourself a place in the symposium so that you do not miss anything from this particular event. All right, so let's not wait and waste any more time. With all my good wishes, would like to introduce the host for today's session. Today's session will be guided and directed by Ms. Ruthvi Vaja, who is the Vice President of the Sci-Omics Student Club. So from this point of time, I would like to hand over the stage to Ms. Ruthvi. Ruthvi, it's all yours. Please take away.
Hi, Shubhajit. Am I audible?
Yes, you are audible.
Oh. A very good evening to everyone present over here. I'm Ruthvi Vaja, the current Vice President of the Sci-Omics, the Student Club for Bioinformatics. Today we have gathered here for one of the very auspicious sessions of the Sci-Omics Club. Today we would be discussing about how to ace a first presentation. So Shubhajit Barua, who is the current president of Sci-Omics Student Club, and I myself, Ruthvi, I'm the vice president of Sci-Omics Student Club. Our community consists of more than 1,000 plus students, 20 plus researchers and clinicians, and faculties, mentors, and peer reviewers to guide us about how to move forward and how to enrich our student community. What do we do at Sci-Omics Student Club, and how are the topics that we take are guided into ways to make students easily understand the basic bioinformatics? So we cover topics of infectious diseases, microbes and microbiomes, precision oncology, plant sciences, neuroscience, and together with these topics, we help students learn how to apply big data in biology. One of the main goals of Sci-Omics Club is to help students clear concepts of bioinformatics. Not only this, but we also help them to learn about basic insights about the tools of bioinformatics and how to apply in the real life research projects. We have brainstorming sessions so that we can discuss with each other and get to know the hypotheses that one can work on and continue to develop as a project and then into a research paper. We have also a new very evolving field of how to apply coding and skills of programming into bioinformatics. All this together helps students and this student community to understand why bioinformatics is used and how can we apply to the clinical problems and other stuff. The main goal of OmicsLogic company is to provide training in bioinformatics, and this helps students to enable independent research, which is obviously guided by mentors and peer examples. We are here creating a developing and a growing community with shared passion for data-driven research and appreciation of citizen science. We help students, clinicians, and faculties of all backgrounds to develop novel and independent research projects using the latest technology and bioinformatic tools in the current data science industries. One of the most happening things is the Omics Research Symposium 2022 that is on March 31st and April 1st, 2022. In OmicsLogic Research Symposium, we would be discussing various aspects of machine learning, artificial intelligence, bioinformatics, astrobiology as one of the most evolving fields these days. We have academic experts from Amity University, Georgetown University, Tauber Bioinformatics Center, Premas Life Sciences, Faba Academy. We have speakers from Google. We have experts from networking partners such as LBRN, H3A, Bionet, and further on. Now, one of the main goals that students faces while going after one university to another is certain kinds of gaps that they need to fill on. One of the most important things that the universities look for into students are the students capable of doing cutting edge research? Just doing any part of research does not suffice for you to get into a good recognized university. So for that, you have to do a research which stands out and which really makes an impact into the scientific community. Not only this, but universities are also looking for placements for students. So they want the best of the students, and the best of the students should know that what is the current technology, what are the researches that they can apply their bioinformatic tools and knowledge into, and stuff like that. Not only doing research and publishing papers, but how well you can translate your research into the clinical aspects or into the translational medicine aspect. So that is taking your research from the lab, that is wet lab or dry lab, to the market. Now, another question that comes up when applying for any recruitment, dissertations, or internships is, what are the companies looking for? So the companies are looking for students to provide internships, trainings, dissertation opportunities, and further on. They are finding potential employees, both they can be young professionals, middle-aged learners, or expertise, and they want their audience to outreach their programs, spread their name. And in order to do this, one has to be self-sufficient. One has to be involved in cutting-edge research and should be aware of the latest technologies, codings, programmings, and data analytical skills that are available these days. Coming to the most important part, how can you connect this all qualities and what part Omics Research Symposium would play? If you are part of Omics Research Symposium, this platform will bring you to a group of experts, reviewers, professors, and postdoctoral fellows from one of the most recognized universities in the world, from both academia and industry. You getting an exposure to these experts will not only help you recognize where do you want to go, but it would also help you to know that what the actual real life world research demands right now. You would be exposed to a larger audience and peers so that you can know that where you are lacking or how much far you need to go. This would help you define your skill set, and defining a skill set was the main important part of any person's CV or resume. You can have an opportunity for networking and connecting and building your own professional network, which is in demand these days, because as a researcher or professional in academia or industry, one of the most important qualities that you must possess is networking, talking with people, getting to know their scientific research, and further on developing connections, which could be fruitful for you in your future endeavors. So what's unique about OmicsLogic students? So our students belong to top universities and schools with whom we are in constant communication. Those universities include Amity University, Nebraska, LSU Biomed, NIP, Georgetown University. Our students also have a fine understanding of basic bioinformatic tools, complex bioinformatic tools, and the bioinformatic concepts so that they can apply this knowledge to real-life independent research projects. Such courses introduce Python, molecules, biomedical, machine learning, transcriptomics, genomics, and further on. Not only this, but our students also know how to apply these concepts to real-world problems, and they then work on one of the most interesting projects to solve health-related problems or to develop pipelines and certain machine learning algorithms. Here are the examples of poster presentations of two of our students who presented in the Omics Research Symposium 2021. For Omics Research Symposium 2022, we have speakers like Dr. Rita Sharma, who is an associate professor at BITS Pilani. She'll be talking about the emphasizing role of bioinformatics in every Indian research lab. We have speakers from Google, that is Mark Allen, who she would be talking about Google and the NIH Strides initiatives. We have another speaker from Google who would be speaking about Google's partner programs and resources. One of the most evolving field these days is careers in astrobiology. Dr. Graham Lau, PhD from Blue Marble Space Institute of Science, would be speaking on this topic and where you can explore your further careers in astrobiology. And we have Dr. Priniti Mathur from Bioinformatics Education at Amity University, and she will be talking about aspects of research training and how to take the bioinformatics education forward. In Omics Research Symposium 2022, we also have poster presentation competition. These are the examples of students who presented last year in Omics Research Symposium 2021. This year, we are covering topics of omics and bioinformatics, AI and machine learning and data science, astrobiology and space omics, computational biology in biotech and pharma. So for today's session, we have Dr. Raghavendra L. He would be talking about the topic how to ace a poster presentation pertaining to the Omics Research Symposium 2022 coming on March 31st and 1st April. Before we start with this session, let me brief you about Dr. Raghavendra. So Dr. Raghavendran is an accomplished computational biologist with more than 4 years of research experience in subject areas like multi-omics data analysis and data science methods in precision oncology, structural bioinformatics. With a background in biophysics and bioinformatics, he earned his PhD in computational biology from the Max Planck Institute, Germany. He has published many research papers in high-impact international peer-reviewed journals. Prior to joining Pine Biotech as a research consultant and bioinformatics mentor, he served as the lead postdoctoral research scientist at the Jawaharlal Nehru University and worked on an Indo-French collaborative project on developing machine learning models for precision oncology. In the course of his research career spanning over 15 years, he has worked in large-scale analysis of a wide variety of big data, ranging from public-private patient data on cancer biology, plant sequence data, and protein structure data. He is very proficient in using Python and R for bioinformatics. He suffers from a presumably health addiction to computer programming and data analytics, and is familiar with a variety of paradigms and their associated toolkits. He is equally captivated by life sciences, specifically focusing on translational approaches to understand cancer biology and neurological disorders. He has been an enthusiastic mentor to several research students at Pine Biotech and is also experienced in tutoring computer programming courses for graduate students and working professionals. Dr. Raghavendra, I request you to join the session and the stage is all yours.
Thank you very much, Rudvi. After such a nice introduction, I started to feel very good about myself one more time. it's very nice to be part of this students club. It makes me feel a lot younger than what I am actually. And let me start sharing my screen. And I apologize for my camera is not working. That's an ongoing issue with my computer that I have to solve at some point of time. So I want to have first few slides as interactive as possible so that we learn from some of the few examples what we can learn from understanding or from observing some good posters, some bad posters, some posters that can be improved. I must confess right in the beginning that preparing slides for such topic was very difficult for me than preparing some slides for slides related to academic or slides related to educational research and educational information. So let's start with the golden question, right? What is a scientific poster? So I might seem ancient, so because I come from an era where we used to start printing the poster, hang it up with the poster wall, and the exposure of your poster depends upon where your poster hangs, and you have to sell your science somehow. But now it looks like we are hoping we will get back to those golden days in the conferences one more time. But on the other hand, online meetings and online conferences has actually can you see my screen? I think it says my screening, my resume sharing is paused, or you can only see my first
visible.
screen.
You can see the first slide.
Yeah, we are
okay.
seeing.
Yeah, wait. Let me. Let me stop and share again because Thank God I noticed. Yeah. Okay. So this is where we were, and So I was talking about some advantages of having an online symposium, an online poster presentation, and how people started to appreciate the inclusivity or inclusiveness that online symposium and online poster presentation and interaction and network building efforts have brought up to scientific community. So Sci-Omics clubs, I think, is also one of the excellent beneficiary from the whole world moving towards decentralized media, decentralized local, delocalized or decentralized options. So let's come back to today's topic. So what is a scientific poster? So as I said, the purpose of a research poster is to represent the general overview, the data, and most relevant findings of a research project. So typically, research posters will accompany an oral presentation of the project conducted, or will accompany some presenter who is going to talk to you or who is going to present and explain the details of the scientific poster, details of various sections involved in the scientific poster. So that is what I meant by oral presentation. But it should also be able to independently represent the research. So if someone wants to scan through the poster and understand understand more about the research and understand more about the terminologies and the research and their findings and their conclusion in general, the poster should be able to hold its ground on itself. So on and all, an effective research poster actually must provide a clean and a very consistent layout, and it should actually emphasize the research question that we have, and it should actually highlight the important results with the using or with utilizing or with the help of useful and purposeful graphics and visuals. And it should have readable and accessible text. It should include citation wherever it is necessary. And also it should most importantly include your contact information, because posters, scientific posters, are an excellent way to promote scientific collaborations. So they give you a very short abstract of your long research career and also a long research project that you are doing. And if somebody is interested in collaborating with your efforts, then they should find a way to contact you from your research, from your poster. So we will start with the good thing and a good example of what a brilliant poster will look like. So you start observing what a good poster is. A poster can be first in a landscape format or in a horizontal format, wide format, or it can also be in a vertical format. I will give you some examples of the vertical format. So what do you see immediately from this poster? It is organized very neatly, and it has readable sections included, and the alignment and all the other things are very precisely organized, and we have independent sections, so that will help us navigate through the poster. But again, this is not a scientific, it's partly a scientific poster, but it's not a very great scientific poster because we don't know how to navigate this poster. We start with the introduction, obviously, because our innate scientific mind always starts with the introduction. And then should I proceed next to food finders, or should I proceed next to pilot study? Right? Should I proceed down, or should I proceed right? So these are these are some of the organizational questions that you should be keeping in mind while designing your poster. So even in brilliantly conducted study and brilliantly presented poster, we can find some ways to get lost. And this is another example, not on the right side, but on the left side, of a poster that is very neatly organized. And being able to make each and every section stand out independently will help you in organizing these posters, readability and navigatability. Okay, so these two slides are there. That a poster can be horizontal and also can be vertical. In the era of physical process of printing out the poster, and hanging out for displaying the poster. There was a, or there still there is a slight disadvantage in handling vertical posters, because most of your juicy part of your research, like the results and conclusion and discussion, are going to be in the lower half of the poster, for which scientists and other people has to stoop down to read it. But now with the digital era, I think more experienced people in web development can confirm me, but the vertical format is most preferred, because you can align these vertical format very neatly in a web page. And towards the right, you have a poster that looks like it has been built in last 15 minutes. So that's a very badly organized poster. It doesn't sections. It has very improper use of fonts, and not very clear readability is being given here. So we will start looking through some of the examples of these posters, and then start building our own formula for how a good poster should look like, and what we can learn from very neatly presented and good poster. So after these two introduction slides, so I'll let the audience decide the quality of this poster. If you want to have this poster, or if you come across this poster, would you tell this poster as excellently organized, and would you give this poster a positive feedback? Please update me on the chat. Quick. Let's go through it. Let's have it a little bit interactive before I go on and on and on about this one. Okay, next. Okay, we have contradicting opinions and some more. Okay, too much text. Yes, we have contradicting opinion, too detailed for me. Okay, but wordy. Yeah. So I think if I can summarize, people like the organization part of this poster, but they dislike the amount of text that is present in this poster. And exactly that is the case, right? This is not an example of an excellent poster because of several reasons. And those reasons are not limited to organizational or more words, this one, right? It's generally about, first about amount of words. Yes, that is right. Too many words. And there are alignment problems. There is too much information into very small area. And the placement of logos is, that's a personal opinion, but people wanted some people want some photos, and some people do not want some logos and etc. So yeah, some of the critiques in this part of these particular poster can be debatable, but in general, it has a lot of data, it has a lot of words, but so that is why it is very difficult to read, and it is very difficult to follow. So another example of a poster is this poster also has an excellent organization, as I talked about. So you start reading from the background, and you go to the present study, and then you go to the methods, and where do you go? You go to the right side, or you go to the left side, and how it is organized. So except for this specific section and some of these sections, acknowledgements and references being being presented with a very small font size can be forgiven. But there is one part of the result which is given at a lower font size even than acknowledgement. So that is absolutely wrong. So it is very difficult to read these sections of the poster. So you have to prioritize, and you have to identify what do you want to present in a poster. So so we learned two important things. One, the poster cannot be too wordy. Two, the poster has to have an excellent navigational or excellent purpose of directing the user, or directing the audience to specifically where we want them to go, or where we want them to focus on. Okay, so what will you tell about this poster? Is this poster a good poster, or is this poster can be improved a little bit more? We will come to that, Yamna, and yeah, we are going to discuss that in detail. We are just going quickly going through what we already know about the poster, and from what we know and from what we observe, what we can conclude and what we can take away. And then I'll go in detail about different sections of the poster in the following slide. So what do you see in this poster? Is this poster a good poster, or is this poster can be improved, or is it working for you? And yeah, please, yeah, please let me know. So I have a comment which says, can abstract be removed? I will talk about abstract in the following slides. Excellent questions already. So people are getting into the group. So some more, come on, more. Can you follow this poster? What can be improved, or what can be added upon, and what can be removed? And is this poster generally working for you or not? Yes. Okay, excellent. So I got the bullseye right in the second or third comment. So it has a lot of graphical information, that is correct, but correlating text that infers or that actually gathers the results from the graphical output or from the bar graphs is missing. It gives a lot of bar graph information, but what do we read from this bar graphs? That is not very clear. Even in the this is not even from the figure legends are also not clear. So we have to go to conclusion and then look for a search for those specific comparison and come back to conclusion, which is not a very good navigational option. But in general, this poster is organized pretty well. It's almost there. It's close there. It has introduction. It has methods as pictures, so that that it's not too wordy, and it has hypotheses also. But if possible, we can improve this poster by reducing or by completely removing the abstract, and by including bullet points of results along or next to each and every one of those comparison or bar plots that are that is being displayed. So yeah, let's move forward. So from what we have already observed, so let's start summarizing what we have learned so far from couple of important posters and what we have learned. Usually eyes read from top to bottom, and for most of the people eyes read from left to right. So a column format will be extremely helpful, and all of or most of us would have enjoyed some comics in our younger age. So if it is not explicitly numbered, we go from left to right, and the long columns will be read first. So that is how a poster will also is also like expected to be organized. Okay, and use sections and use small boxes or small sections of subsections and sections of information, so that people can get a feeling of navigating through your poster. If your poster is an expanded version of your paper or of your abstract or of your profile or of your proposal, then that's not a good poster at all. Even if it contains a lot of pictures, it should not have complete text information. So people have people can follow or read from anywhere between 12 to 16 words in one line on a horizontal poster, and even less than 10, 12, less than 10 words on a vertical poster if you use in one line. So use sections and headings that will give a navigational aspect to the posters, and use a lot of graphics instead of text, but you should accompany these graphics with what do you learn. It's actually your research, and you cannot just put on some of the plots like we saw in a couple of slides ago and expect the user to make the connection. You should make the connection and tell them where you made this connection from by displaying the plots, by putting up or by including the graphics. So these graphics can be comparative plots, these graphics can be pictures of the experiments and results that you perform, and these graphics can be results of the analysis that you calculated. So we should definitely include high resolution pictures. That is extremely imperative in this age or era of digital poster presentation. And we should label each axis with units. We should include legends for color legends or shape legends for different graphics plots and comparative plots and comparative graphics that we used so far. So if you want to include text, how should you include text? We should include text in short phrases and in, if possible, in bullet points. If you have, or I have also seen, or it is also quite common not to include bullet points for conclusion or specifically for discussion, if discussion is included in the poster. There the aim is to build up a cohesive argument or cohesive speculation of the results that we have. So it is quite okay to have text in short phrases and not long complex statements in the discussion part if you want to include discussion, but mostly conclusions or summary is preferred over discussion in a poster presentation. So we can include text, but at which font size? That's a golden question. At which font size? Can anybody guess? If you had made some poster and if you have some common example of making and developing posters, then you should know what is the absolute minimum font size that you can make this poster, make a poster with. Yeah, 32 is very conservative. Excellent. Yeah, 24, right? Nothing less than 24. This 24 font size on a, I mean, nowadays font size depends upon the font, but on a normal fonts like Arial, Calibri, and on these fonts, then anything between 24 and above, or anything between 24 to 32 for your normal text should be good enough for the for the participant or for the audience to read it from approximately 4 to 6 feet far from the poster. That is the aim. So anything smaller than 24 is not going to be very legible, and some texts like references, like acknowledgements, like funding acknowledgements, and like special cases, these can be a little bit smaller than 24 to save or to have some space realized in the poster. So headings must be around 70, 72, around this size, because headings are the one, or titles and headings are the one that actually pull people from far away. So it has to be right at that number, or so that it is legible and clear. So you should learn a little bit about visual hierarchy when you're going to present the poster. So spend some time in reading this slide. So what do you see? Do you see all that is given in this visual hierarchy? So you can form a priority, or the priority in which you are reading these different texts all by yourself, right? So you will read bold text first, and you will read brightly colored text first, and you will read the text that is presented in a font that is not very difficult to read first, and you will completely skip if the text mingles, or if the text is somehow similar to the background color, or if the text is too light. It's very difficult to read it even if you skim. So you should keep this in mind, and make your posters, or make the text in your posters readable, and by mentioning this, and you cannot just make each and every line or each and every word of your poster to be bold at 32 font. So that is also not going to be very good. So contrast is very important. Which point that you want the participant, or that you want the audience to highlight. So those things you should actually decide when designing a poster. So there are some font styles which are extremely pleasing to read from distance, and some font size which are not very pleasing to read. So you can use some. I will also give some more cheat sheets about some font combinations and color combinations later. So which you can explore for the possibility of using different font size, because we don't want to end up using the same font size for everybody. If you have the freedom to use different font size, explore. I have a question on which is the best software to use to design a poster. Nowadays, excellent posters can be designed on Microsoft PowerPoint itself. So PowerPoint has a capacity to design excellent posters. You can leverage the use of PowerPoint. I will talk a little bit about some important technical aspects of PowerPoint later. I think I have it in 1 or 2 slides. I will come back to that point later. Okay. And if you have to use background, don't use distracting background. So use a background that is not distracting. The background should, as the name suggests, should not actually compete with the information that you have. So use a light or fair backgrounds with the dark letters. That seems very easy and legible to read, and not very dark backgrounds overall with the white or with the bright letters. That is not very much preferred, because you have to change everything from the plots that you make, from the table that you make, everything depending upon your choice of the background. So generally light background or lightish background with a very soft gradient gives a very pleasing look and feeling, and warm welcoming feeling to your posters. So a typical organization of the poster depends on what you want to include and how do you want to include. So you can have a lot of flexibility in the poster depending upon what do you want to add, and how your study dictates what you want to add. So in an academic setting, when you want to present a poster that involves multiple authors, and you can stick to the order of the author, but you have to highlight the author who is presenting the poster there, right there. Title should not be more than 2 lines. At its most, it should be 1.5 lines or 2 lines. It should not be more than 2 lines. If it is more than 2 lines, then that is not people lose interest in the title itself. So it should not be more than 2 lines. And then you can have sections organized as you wish. You can have an abstract, you can have introduction, you can have methods and results, and you can have multiple results and conclusion and references. So you can change up these sections. These are not hard rules that you have to stick to these sections all the way always. So for example, if my paper, or if my work is not about it does not involve a lot of methods, then I can change up or use a lot of area for results to discuss results, and I can eliminate the abstract completely and then move the methods down here and do all sorts of things that I want to that I want in my poster. So it is in fact my poster, and within the given real estate, you can change up the organization of the poster to suit your data and to suit your study best, right? Keeping in mind the flow, keeping in mind the readability, and keeping in mind the general template that you want to stick to. So where do we begin? So what do we how do we start building a poster? Okay, where do we begin? So we have to first find out, is it okay to use some sort of arrows that do not look or to keep flow with the heading? If you want to if you want to navigate, if you want to help in the poster navigation, you can use numbers. 1 introduction, 2 methods, 3 results A, 4 results B, 5 conclusion, 6 references. That is better than giving arrows, okay, than using arrows for the flow of headings, because depending upon how much text you have and how much information you have, you have to make some sort of compromises on the navigability and readability, and also general organization of the poster. So let's come back. So when you have completed your project, or when you have your project ready for most of the analysis ready and most of the interpretation has been going on, so you have to start asking, what is the one most important finding that from my research project that I want to share with people. If all the posters can include just only one line, what will that one line be in my poster? So that is the most important question that you should answer from your research. You should write it down and write possible candidates for this specific question, and then come back later, because it's we work very well when we work subconsciously. So come back later, and then you start precipitating the order or the priority of extremely interesting finding in your research project. And then second one, how can you actually use different visual elements like charts, graphs, pictures, and flow charts that is going to help you share that one interesting finding with the conference attendees. So everything depends upon that one hero, that one highlight, right? That one protagonist that you want to highlight, that you want to support with the use of other tools. And what it is just not sufficient to add everything in your poster and then take a break and go to your take a break and wander off like that, right? You have to think about, what should I talk? I obviously cannot read. If you are just going to read what's in the poster, then why are you there? People can read themselves. So you have to tell the people what's in the poster, right? This is very important in the era of digital poster presentation, because people, including the jury members and other participants, have access to your posters all the time. They can look at your poster asynchronously. They can look at your poster while some other non-interesting session is going on. So if you are just going to read what's in the poster, then that is not enough in this era. So you have to find out, you have to write it down, or you have to narrow it down to what is the information that you want to convey. Like the information should be about how did you start your experiments, or how did you start your research, so that all of your research focuses on finding that one most important thing in your research project. So that if you start building up like this, then you will start forming a story. So when a story builds, then you will have a lot to discuss about your poster. So in the poster context, the 3 M's that we should keep in mind when creating a research poster is regarding message, medium, and materiality are the 3 components or central components that should come together so that you can make, or you can form a context for your research posters. So within these 3 messages and mediums and materialities, so you have to first narrow it down about the message, right? Who is your audience? Is your audience all of your audience are senior pros in the research, or all of your audience, or most of your audience are your peer members, like fellow students and senior students and junior students, etc. So what are you trying to communicate to them? Is your area is one of the popular areas where everybody has some introduction or background to, or do you have to spend some time in explaining your background or your area, the project's background, a little bit to the audience? Those are the contexts that you want to keep your eye on, or keep it in your mind. So again, as I mentioned, what is the one most important message that you want to convey with your research poster that you have to identify? And in the medium, where are you presenting this information? Are you presenting this in an asynchronous format, like and also synchronous format, like in person or in a virtual conference? And how will you be sharing your work? Will you be recording? Will you be recording a short message about your poster, a presentation about your poster, or will you be presenting it live to people? So and your script or your medium of explaining or discussing your poster will change depending upon what you are, depending upon the medium that you want to present your results or present your posters on. So in the materiality, it's about how can your research be applicable beyond the context of, or beyond the beyond the restrictions of your field, right? What is the importance of this information? Let's say, okay, I found out that by benchmarking something like 50 different machine learning algorithms on a simulated data set, I found some of the algorithms perform better. Is this a good work? Maybe yes, it is. I spent something like 1.5 years to do this. Is this important work? I don't know, because who cares about simulated data? So is this work interesting to many people? I again don't know. Unless and until you apply this to real world data, nobody is interested in simulated data for estimating the accuracy of machine learning algorithms. So lot of different things should go into deciding that what one most important message and how that important message can be packaged so that you can show it to your fellow peers. Most of the people who are going to read your posters are not going to be experts in your research area. So they are going to be scientists, and they are going to be on their way to becoming a scientist, but they might not be expert in the exact analysis methods or in the exact processes that you took to obtain this research. So you have to decide upon the importance of information and how your research topic is going to be presented in a relevant way to your audience and in the field of study. So while moving on, let's talk a little bit about appearance or technicality. So apart from this, from these generalized context of identifying what to present in the poster, you will find lots of what to do, what not to do in generally I mean scattered all over the internet about the posters, right? So I will give you pointers in what to add in the posters. Then you can use those guidelines and pointers to fill rough draft so that your poster can come to shape instead of just talking about a generalized concept of what a poster should look like and how do you identify your research question and how do you identify your most important message. So in the appearance, your poster appearance, it depends upon what do you want to how do you want to organize the poster. You can organize the poster as horizontal or vertical, and as a 3-column, as a 4-column. Anything more than 3-column in horizontal or even in vertical is not going to be very legible, I will tell you. So if it's vertical, it's 2-column, and if it's horizontal, it's 3-column. So inside the poster's appearance, some general acceptable and not acceptable patterns are: you can have images in the introduction, but that is going to eat up some of the real estate in the introduction. So mostly that is not preferred. But you can have, if you absolutely want to introduce some of the existing research or existing conditions, you can have introduction, right? Introduction is all about why did you choose to study this topic, and why did you choose to perform this analysis in this specific system. So introducing the system, introducing your study, introducing your data is what introduction is for. So methods can be flowchart, or can be some sort of graphics art, and can also be bullet points. So it depends on what you want to add, but mostly preferred is methods can be something that you can relate that connects a sequence of events or sequence of designs. So in the results, obviously you should have a lot of figures, line graphs. They are common, excellent. And then in discussions, I just spoke about discussions being not bulleted, but it's often bulleted. But you should mostly conclusion and summaries are preferred over discussion, because you will often not have enough space to discuss enough things in a research poster. So we spoke about neutral backgrounds. We spoke about not very strong highlighting backgrounds and distracting backgrounds, and use some good white space in your poster, and don't cram it like what we have what we saw in the first few slides. And a general size guide or number guide for how much information that you should add in your poster is about like this. The titles should not be more than 1.5 or 2 lines, and the introduction is about 250 words, and in aims or specific aims or objectives of your research work should be less than 100 words. It should not go on and on. More often than not, the hypothesis or the objectives are the shortest or smallest portion of your poster. And again, materials and methods should not be more than 150 or 200 words. Yes, you can have my slides. Yes, so should not be more than 200 words. And if it is becoming a little bit bigger, please try to condense it into some sort of flow chart or some sort of flow diagram where people can follow. You don't have to explain everything. You don't have to explain how you are doing Western blot. You don't have to explain how you are doing PCA plot, right? These are the things that people can understand. So you don't have to explain how you did it. You just have to find out a precise balance between what you did and explaining what are the methodologies that are not very common, or if you have used any specifically different methods that you have to mention, that you can mention. And your results should always accompany the figures, and it should not be too wordy. Results should actually tell what you see from the figures, and results should not have conclusion, right? You see what, I mean, you have to tell what you observe from the figure, and you can tell what you find some of the implications from those observations, like A is greater than B. So why is that A? Why do you think A could be greater than B? Or what is the effect of A being greater than B? A is greater than B, and this A being greater can affect or can result in C being greater than D, and etc., etc. And A and C being greater in the samples, this set of samples can be due to this set of samples being recorded under this condition. They are from this disease sample, etc., etc. And A and B are implicated in these diseases in few of the prior research that is already been performed. So this is how you can expand on your research, but not more than results, not more than 200 words, something like that. Right? If you have to include acknowledgements, you can include acknowledgements, but very little, because sometimes when you are doing funded research, it is expected that you include some acknowledgements of those funding resources. So you can just mention their logos, and you can just say acknowledgements, those logos, that's fine. You have to include references. That is one place where you should never ever compromise. You can make it however small you want to make it, but you have to include references. You obviously you cannot perform any research without referring to some of the prior researches, prior work. So you have to include research references. So if you want to go one by one, we can go. So title and abstract. Most of the posters will not have abstract, because my personal understanding and my personal view is your poster is the abstract of your study. So you don't want to have another abstract of the existing abstract. So you can use those section for explaining a bit on the background, or adding some more conclusion, or adding some results part. And sometimes you might be stuck in some organize or in some with some necessity that you have to include abstract. Then you can have the abstract at its minimal, at its absolutely a minimal requirement. You can have that, and you know what to include. You should include the question. I mean, you all of you would have submitted abstract to the symposium that we already have, or most of you would have done it. So you should include what are you researching. You should include what the method that you use, and what did you observe, and what are the implications of those results and observations, and what are the major findings and conclusions that you. If you start answering all these questions in two lines, you will arrive at a 200-word abstract. So that is how abstracts are formed. And after this, you have to improve that paragraph for better flow, better highlighting, and etc., etc. But this is how abstracts are written. So introduction or background should not be a copy of your abstract. It is completely separate. It actually tells you what are the current research that is going on, that is related to what you have researched, and where is the gap. The main flow or necessity to introduction or to the introduction piece is to introduce to the audience how or what has been done in the system that you are working, or in the field that you are working, and how these two combine to present you with a gap, that you are addressing that gap. And So when you do this, you will arrive at a purpose or a hypothesis or a specific aim for your work, for your project. So that is where introduction to culminate into. So you start introducing about the general aspect. If I'm working on cancer genomics, I will start introducing about cancer being the deadly disease, and then slowly focusing on which cancer that I'm going to focus on. And then second, up in that cancer, what is the model system that I'm working, that I'm taking the data from, that I will focus on. And then in the third paragraph or in the third section, how these two combine, how I'm using this model system to answer a general question about the cancer biology that is not answered so far, and what are the specific methods that you use to do this, and what do you find. That will finish up your introduction, and then this comes nicely, or this goes nicely with a very specific hypothesis or aim that you are going to formulate at the end of your introduction. So that should naturally flow into the problem statement or the aim of the project or the hypothesis of the project, depend on what you are doing. So so that is it can be embedded within the introduction, but it adds more value, even however small it is. It adds more value if you add it as a separate section. So sometimes many people will just skim only the aim of the project or specific aim of the project, so that if they find whether that goal or that objective pleases them and interest them to proceed further to read about your poster. And then comes methods. We spoke a little bit more about these methods. So methods should not be like this or like this. It can partially be like this. It is acceptable because this tells some story, and this gives some image, pictorial representation and something related to that. But materials and methods should not be like this. This is like I copied and paste from publication. So your poster is not a copy paste from your publication or your draft. So you don't do that. Yeah, sure, Uzma. I think Sci-Omics will take care of that. So method. This is an excellent method. Maybe a couple of lines added to this will explain, will make this method complete. And you can have, if you want to have lot of text in a method, please use bulleted points. That will declutter a lot of your clutter in the text. You can have short, strong statements, add bulleted points. Instead of making a statement like I took this data, submitted, made this pre-processing and submitted to this analysis, you can do the data was this analysis was performed on the data, finished. If people want to know more about that analysis, they can look it up. And then comes important part, images, charts and graphs, and which you should you will include in the results section. I think I was intended to add this after this. Okay, but that's okay. We can take a good look about this. So you remove unnecessary borders, you remove unnecessary backgrounds, remove unnecessary lines, and remove not very clear labels and markers from your plots, which are default option of Excel. Don't just make a plot in Excel and then copy and paste it in your poster. It is going to come up with the bluish gray background, and that's not going to look very good. So yeah, use solid colors and flat styles, and not very bad table. So this is an example of a table that you often see in Word or in Mac pages, but it's not a very good table. It's a very bad table. Use table like this. And tables, you don't need a vertical line for the tables. So you you don't need horizontal lines for the tables. You can have vertical lines, but something like this looks really pleasing and really good. So most of your charts should be accompanied with something about what you perceive here. Whether you perceive that the blue is consistently higher in all the comparison, or the blue is higher in some of the comparison, or the red started increasing as the comparisons or the categories go along. So you have to find what is important finding from your poster, and then you have to add those lines precisely next. So results will form bulk of your poster section, right? In terms of occupying the real estate, and also in terms of material, and in terms of stuff that you want to put in, images and text that you want to put in. More often than not, you will struggle with what to have and what to leave. So it will change depending upon the field that you are working on, but mostly it is going to be one big column, and this is going to be the section that summarizes your important findings of all the analysis or important analysis that you did. And you have to draw you have to make specific or make important implications and conclusions from this, and how it leads to your one important finding. So that should be highlighted, and how different results combine, so that combine to to make you conclude that one very big important finding. That is where your result should lead to. Excuse me, if it is possible, unless and until it is necessary, do not use long tables. If you use long tables, it's very difficult to read. It's very difficult for people to follow, and it's very difficult for you to explain also. If you can concise, or if you can condense that table into some sort of pictorial or graphical representation, that is excellent. But if you absolutely must have tables that is not more than something like this, not more than 5 rows and 4 or 5 columns. That is the maximum that you can have a table. Anything more than that is going to clutter, make your results look really cluttered. And you can have something like what people do in physical poster presentation is have a handout. In those handouts, they will have appendix where you can paste a lot of big tables if you really want. But now I think in the online and in this era, in the digital era, they do not give the possibility or give the option of adding some supplementary materials, and that is to be considered for many people. So what is a general rule? A general rule is one paragraph should accompany each image section. Like these two are one image section, so what do you learn from these two images or from these two comparisons? That is one small section. And these two are one small section, so you should have something written. What do you learn from these two images? So if you have all of these, then it's very difficult to refer to where and what. So if you write even a big paragraph about referring, first go to A, I conclude that from A, and go to B, I conclude that from B, go to C, and C implicates this, and D gives you important results, that is very bad. So you take this as a separate section, and within those first two, add what you learned from first two, and the next two, add what you learned from next two, and the next remaining plots, add you learn from what these plots tell you. These plots can be arranged in the algorithmic, or in the what do you say, in the order in which you did the analysis, or this most likely or most preferred, this has to be arranged in the story, in the order of the story that you want to tell. It can be very well happen that you performed one of the key analysis in the later part of your chronological order, but that becomes one of the key component in first few identification or first few sections of the results. Like yeah, so those can also happen. And you found out that you have to fill some of the exploratory analysis that you have not performed earlier. So if you perform those exploratory analysis at last, that not be mentioned in last. So that has to be, exploratory analysis has to come in the beginning when you are explaining your data. So these sections plus you can add something about the data if you are mostly for biological or bioinformatics people who are taking publicly available data to perform this analysis. So your methods section can include data. So that is materials and methods, right? So material include the data. So you can actually talk about the data set that you downloaded, the data set that you chose for, the little bit about the data set, where to find the data set, and how did you get the data set, what this data set generally have, like a very small pivot table and very small table in terms of number, like these many normal samples of tumor triple negative breast cancer is compared with these many treatment sensitive samples of triple negative breast cancer. So that you can have it in the material section. I wanted to tell in the material section, I forgot that. So finally, your summary and conclusions. So it is preferred you have summary and conclusions like this, like bulleted points, so that it flows, and people tend to remember and keep a lot of things in their mind when it is presented as a list or as a bulleted point, and not as a summary of conclusions in like what you obviously see in publications. Like your summary should be one result, or that most famous result that we talk about, right? That take home result that you want to that you want the viewer, that you want the participant, or that you want the audience to take home with. So you mentioned some specific game. Whether or not did you succeed in answering those questions in the specific game, you should always relate to that. Your result or your conclusions or your summary should always result to that. If my specific aim is to if I have stated that I want to find out if genomics data can actually help me categorize the locations of head and neck carcinoma, then I should tell whether or not I succeeded in this effort. Like I tried it did not work, I need more data. This can also happen within the realm of within the availability of percent data. I concluded this I can say with 50% or 60%, 70% certainty that I can sufficiently categorize head and neck carcinoma from different sources based on gene expression data. So always your primary result in the summary or conclusion to relate to the hypothesis or to relate to the specific aim that you have mentioned in the paper, or sorry, in the poster. Use similar wording. If you have used something like transcriptomics data, aim is to identify whether transcriptomics landscape can explain breast cancer subtypes, then you cannot add I used RNA-seq data to understand, or these RNA-seq data can actually understand tumor landscapes or tumor subtypes. So make it in such a way that people who are not familiar with the terminologies in your field can relate. If you use transcriptomics data in the hypothesis, use transcriptomics data in the conclusion. If you use RNA-seq data, RNA-seq pipeline, RNA-seq analysis right throughout your poster flow, use the same word in the conclusion and summary. Don't mix match. There may be multiple words and jargons associated with one specific meaning, one specific data or one specific type. Don't mix match. That is going to be inconsistent, and people are not going to follow it. Okay. And definitely add references can be added in multiple ways. It can be added in different formats. Use the smallest and shortest format that goes along with the space that is being available to you and with the format that is being supported. Definitely add reference. You should add definitely add references to your study. You can obviously would not have been able to do this study without references. So definitely add references and refer those references in the introduction and in when you are relating that in summary and conclusion. Refer those references. Just don't add these references. Do point those references, cite those references in the introduction. So when you say that cancer is the deadly disease, it has already caused 40 million people death in the last these many years, I have to refer that this is from the cancer genomics or GDC or TCGA data. So I have to refer that. You have to cite that. So please cite it. And let's talk about some cheat sheets or some cheats about how to understand or how to go about organizing the poster. As I mentioned, so 4 horizontal organization is the maximum that you can take in a poster organization that is horizontal, and not more than 2 in the vertical format. And you can have different document size like 36 and 54 and 72. Here comes the PowerPoint tidbit that I want to tell you. If you are going to use PowerPoint to make your poster, I think there is a limit of 56 inches on both the height and width of the slide, right? You cannot have more than 56 inches in the height and width of the slide, but you can have proportional sizes. If you want to have something like this, 72 inches, so you divide this by 2. You make 36 by 24, and then you make your PowerPoint presentation and you zoom it to 200. You set the page size before start making your PowerPoint, before making your poster in PowerPoint. Because if you start making your poster in some different free form, then start to condense it into necessary size and necessary page setup, then PowerPoint is going to ask you, should I expand or should I collapse? So that is going to mess up with the alignment, mess up with the font size, mess up with a lot of things in your poster organization, so poster presentation. So don't do this mistake that I've done a lot of times. So first set the page setup and multiple pages of the same page setup in the PowerPoint presentation. You can have multiple drafts in that way, so that you can track the progress of your poster presentation. And then your poster layout, right? What are the good layout and what are bad layouts you can have. If you want to have graphs at any form, right, on plots at any form, these are the acceptable poster layout. Do not use layouts like what they use in newspaper. Do not fill these pages with the text. That is a very bad poster layout, right? And if it's a circular plot or if it's a network plot, if you have a lot of white space around this plot, it does not matter. It has to stand alone. It has to stand alone within the squarish area, within the encompassing square area, that it has to stand alone. You cannot fill this area with the text that people want to read. So people should read. No, this can have legends and this can have information related to pointers related to this specific pie chart or the specific graph, but not the readable text. It cannot have the readable text. So please use a layout that makes sense with the research that you are going to use. And these are some of the basic layouts that you can use. And these are some of the basic cheats for title, for the other font sizes that you have. So somebody asked me about headings. So use heading that is highlighted in a separate section, or do not use heading that flows along with the line of the text. So this becomes very difficult to find out where these headings are. You can have numbers here in these headings so that you can generally direct people to where they want to go. And these are excellent headings. And this is very bad background because we cannot see what's going on. And you have to select your background really very nicely. And these are some of the font size and how those fonts will look like. You can mix match this so that you can have a very unique presentation style for your poster. So so your aim should be to make the viewer easily identify each and every section of your poster and follow the flow that you want them to follow, right? And let's move on. Like some of the colors which will go very well with each other, right? Red background, blue title or blue font will never work. Don't ever do that. It's very difficult to read. You can make one in the PowerPoint and try to read it from like 2 feet away. You will not be able to make out. And similarly, blue background, red dots or red color font will never work. So if you want to use red or green or blue background, use whitish, grayish or bright colored fonts. So those will work very well with each other, right? Very well with them together. So don't just because you like red and blue, don't make them as red and blue. So this is a bad alignment that I was talking about. Don't do this. Even even many newspapers do that. Don't do this. This is very difficult to read, and it will look cramped up, and it will look really messy. So I think, yeah, with that I conclude. I hope you all had a lot of very valuable information and very good direction in how to design your posters. And yeah, I can take some of the questions if you have any, and I can share this presentation with you, and I add some of the references that I use for this presentation in the appendix, and then I can share it with you so that you can take information from this poster or from the referred media that I used to make these PowerPoint slides. Thank you very much, and I hand it over to Ruthvi, I think. Yeah.
Thank you so much, Dr. Raghavendra. This was really an enlightening talk for students like us to understand about how to, actually present a poster. I'm very sure it's very helpful to others as well. So are there any questions in the chat box? I think, yeah. Can you just tell a bit about plagiarism while making posters?
You should not plagiarize. Definitely. You should not plagiarize. That's nothing about that. If you want to quote, you can quote, like many budding scientists or many people like you, when you are coming up, you will struggle to reformat the introduction section, right? If you want to add the introduction section from some quote introduction section or site introduction section from some other paper, you will struggle to reformat that into some other wordings or some other words. So in that case, if you absolutely want to take the exactly same words, you can quote, like I did it, right? Cancer is the leading cause of death. It already killed something like 10 million people. so far. So this as quoted by this source, you can quote. Obviously, you cannot use it for all the lines in your introduction. So for specific things that you can quote, you can do that. But don't plagiarize about. I mean, for about anything, for anything. You can modify it. You should not absolutely copy and paste it. If you want to use yes, for example, if you want to use some of the for example, like the effect of cancer or the availability of big data, if you want to put graphics there, you cite them just right below the graphics. You cite from which website you took this picture, or you can modify that picture by you, right? Modification is not cropping. Cropping is not modification. Taking a picture and then cropping their logo is not modification. Modification is that taking the picture and extracting the data from the picture, making the picture again in your computer is what is modification. So that is not plagiarism. That is modified picture, that you recreated that picture in your own computer based on the existing data. So that is not modification. So some of the pictures are available freely, Creative Commons license, like Wikipedia. So those you don't have to feel bad about. You can just mention it and then just give proper citing, like Wikipedia, this link, finish. So you can do that. So don't plagiarize. Try to change it up a little bit, rephrase it, change it up a little bit so that you don't end up plagiarizing. So a good writing flow is, if you want to absolutely take some section, some introduction from this paper, some introduction part from that paper, another introduction from this paper, you copy everything and then put it in a Word document and try to write it, try to type it, the same word, try to type it again. So when you are typing it exactly same word, looking at that and typing, you will start forming your own sentences and your own statements, because you are the one doing research in your area. So by forming your own sentences and statements from the idea that you just took from other papers, then you can condense and you can completely change the form or change the text of the change the text so that it is not plagiarism anymore. That's how we do. That's how we write some of the introduction part. Okay. And any other questions?
I hope, Bithya, that answers your question. Since we are running out of time, we might take some questions after I finish this introductory part. So in the spring semester, OmicsLogic in March 2022 had some of the great workshops such as NGS single cell data analysis and single cell RNA project support, detailed analysis of cancer mutations using next generation sequencing, and the most exciting and upcoming event is the Omics Research Symposium. You can scan the QR code over here to register for the Omics Research Symposium. I heartily welcome you all for attending. It is a free to attend event for students of all levels, faculty, professionals, and experts of all backgrounds. You can stay connected with us by joining us on Facebook, Instagram, LinkedIn, YouTube, and Twitter. It would be really great for you if you can scan this QR code and fill out the feedback form so that we can come back stronger and help you to get the sessions that you already want. I would like to thank Dr. Raghavendra for his insightful talk once again. We were very happy to have you over here. Thank you so much, sir. And with that, I would like to end the session and pass it on to Shubhraji. Are you there?
Thank you very much, everybody. It is my pleasure to interact with you all, and it's as I mentioned, it feels really, I feel younger when I'm interacting with you all, with all your bright minds. So thank you very much for having me. Thank you.
Thank you very much, sir. Everyone, that's the end of our meeting. Thank you for joining, and have a good weekend and have a good night. Thank you. Bye.
Thank you, everyone. It was lovely to have you all here. If you have any questions, please feel free to email us in the email ID mentioned in the chat section. See you again soon. See you at the symposium. Bye. Take care.